Good morning. I would like to welcome all of you who've dialed into our investor call with the management of Coya Therapeutics. My name is Kay Nakai. I'm one of the senior healthcare analysts here at Chardan. As call participants are aware, our focus at Chardan is on identifying companies that can generate exceptionally high long-term investment returns by creating shared value for society through disruptive innovation. Joining me on the call this morning from Coya are Chief Executive Officer and Chairman, Dr. Howard Berman, Chief Business Officer, Dr. Arun Swaminathan, and Chief Medical Officer, Dr. Fred Grossman. Before we get started, just some compliance issues. During this call, we will not be discussing Chardan research. Any discussions about research should be coordinated between a participant and their respective salesperson at Chardan. A recording of this call will be made available to clients after the call. By participating in this call, our speakers attest that they have made Chardan aware of any potential conflicts, and they will not discuss any material, non-public, or confidential information that they are aware of that may breach their legal, regulatory, or fiduciary responsibility to any parties. With that out of the way, for anybody in our audience who'd like to submit a question to ask of management during the call, please submit it to me at my email, knakai@chardan.com. With that, let's get started. First, gentlemen, thank you all for joining us today. Maybe to start, Howard, can I ask you and the management team to spend a couple of minutes to provide some background and a brief overview of Coya for investors who are new to the story?
Absolutely. Good. Well, what I'd like to do very high level today is to discuss Coya's unique approach to using combination immunotherapies to target neurodegenerative diseases. And we focus on inflammation. And I say combination therapies is because now I think people are realizing that in order to slow or stop these diseases from progressing, we need to target multi-pathway approaches, much like in oncology and viral diseases, where in many instances, patient survival has been transformed. So we, as a company, high level, have been public for about 1 year. And I'm happy to report in that 1 year, we've been able to advance our portfolio, our combination biologic Coya 302, and we're gearing up to start a clinical trial, a randomized double-blind trial in ALS. We're expanding our portfolio, and we've been able to execute what I consider to be a transformative licensing transaction, bringing in non-dilutive funding and capital, and that was concurrent with a PIPE transaction, which was done at the market price, and it was done with no warrants and no toxic provisions. So I think we're uniquely positioned. I'll have Fred Grossman, our Chief Medical Officer and President, talk to the program, the high level, and then Arun later on will discuss our licensing transaction. So Fred.
Thank you, Howard. Coya is really a disruptive company that's focusing on regulatory T cells to reduce inflammation. It's important to realize that inflammation plays a critical role in neurodegenerative diseases. And by focusing on regulatory T cells, we enhance Treg function and number to reduce the downstream inflammatory pathways. In fact, we are following the discoveries of two key individuals who are on our scientific advisory board. Shimon Sakaguchi, who's a distinguished professor at Osaka University, who essentially discovered Tregs in 1995, and Stanley Appel from Houston Methodist, who essentially pioneered the role of Tregs in neurodegeneration. And as you'll see, our focus is on using our biologics portfolio, and as Howard mentioned, 302, which combines two biologics to treat some of the most severe neurodegenerative disorders, initially focusing on ALS, but also looking at dementia as well as Parkinson's disease. And as you'll see, as we discuss some of our data, we have excellent preclinical and clinical data in patients in ALS and in Alzheimer's disease. We're moving forward very significantly with our first asset, which is Coya 302, which combines two biologics, low-dose IL-2 and CTLA-4. We'll talk about that and why we see this as a pipeline within an asset, first focusing on one of the most severe neurodegenerative disorders, ALS. And in fact, as Howard mentioned, we will be starting a significant trial first half of 2024. And we'll also be reporting on Coya 301, which is low-dose IL-2 in a double-blind, placebo-controlled Alzheimer's study. And we will share the study in patients that led to the Gates Foundation funding that important double-blind, placebo-controlled study. So we look forward to speaking with you about this. And as a company, we have an expanding pipeline that is very significant, that has both near-term as well as other innovative assets as we move forward.
Well, great. Well, thanks, Fred. So Howard, maybe to set the foundation for our discussion about your Treg enhancing biologics, let's first talk about your experience with Coya 101, which was your autologous T cell or Treg cell therapy. And this drug was evaluated in phase 1 and phase 2A clinical studies in ALS patients. What did you learn from these studies about the underlying immunomodulatory properties of Tregs and their potential therapeutic benefits? And how is that informing your current development efforts?
Okay, thank you. So first, we learned that you could take patients' cells from the body and you could expand, you could take dysfunctional Tregs, you could remove them from that toxic inflammatory environment, and you could expand those cells using our manufacturing and make them normal, make them functional, and put them back into patients. And that was essentially the model by which we were going to treat these patients. And we accomplished a phase 1 and a phase 2A study with very exciting results. In the phase 1 study, we showed that putting cells back into patients could either slow or stop the progression. So Tregs were playing a critically important role in this disease. And in the phase 2A study, we also showed that 6 out of the 8 patients dramatically slowed progression. But one of the important things that we realized was that cell therapy is cumbersome, it's expensive, it's complex. The manufacturing in the CMC is complex. And we just realized that also the benefits of being able to upregulate Tregs and to enhance it using off-the-shelf processes like biologics would be much better. I think the most important thing that we learned was that putting live cells back into patients, there is a risk that those cells can convert from a functional Treg back to a dysfunctional Treg quickly in the environment of an inflammatory environment. And in fact, a functional Treg can actually convert to a toxic Th17 or Th1 cell, and that can actually have negative impacts. And so the key lesson here is that inflammation needs to be dampened and tempered down concurrently with enhancing Treg function. And that's why we've decided to add the CTLA-4 Ig fusion protein in combination with the low-dose IL-2. By putting the two together, you create an inflammatory environment in the body that is conducive for the Tregs to survive and to thrive and to enhance, to have durable activity. But we also learned one last important thing, is that oxidative stress is a key driver in addition to inflammation. And we are starting to learn and discover that there are very important biomarkers in terms of oxidative stress that actually not just correlate with the degree of decline of the patients, but their survival. And these are activities that Fred and our team are working on. We'll be working with the FDA on some of these discoveries. We're excited to publish that data and to move that forward in concert with this clinical trial that we're running.
Okay, so great. So Coya 301 is your low-dose recombinant IL-2 therapy that aims to selectively expand the Treg population in vivo in order to enhance its suppressive function and to restore immune system homeostasis. So Howard, can you further expand on its mechanism of action, specifically as this applies to neurodegenerative diseases?
Sure. Well, low-dose IL-2 selectively at a very low dose, much lower than the high dose people have heard about, Proleukin. The low dose selectively binds to the CD25 receptor on the Tregs, enhances the Treg function and numbers, and by doing so, systemically, one can re-educate the immune system in the periphery. By re-educating the immune system and dampening down the inflammation, you can have a direct impact and effect on the central nervous system. And so in ALS, for instance, and in other conditions, Alzheimer's, one of the hypotheses is that the reason that patients are not able to think as well, or they're cognitively impaired, is that there is a dysfunction, there's a heightened inflammation at the level of the synapse, where the signals transmit from neuron to neuron. And that is driven by macrophages and myeloid cells and inflammatory mediators. By being able to remove those inflammatory blocks through the impact of with the use of Tregs, enhanced Tregs, and lowered inflammation, one can actually potentially have patients think better. And one of the key discoveries that we've made, as I've mentioned before, is that in inflammatory conditions like neurodegeneration, the macrophages and the myeloid cells play a really important role in causing the Tregs to become dysfunctional. So low-dose IL-2, while exciting on its own in enhancing Tregs, is even better, we believe, in combination with other drugs that will suppress the mechanisms that will cause the Tregs to become dysfunctional. And we chose many different drugs to combine with low-dose IL-2. CTLA-4 Ig, the fusion protein, was one of the best ones. So the combination, we think, with 301 together with the fusion protein really serves an important role to condition the environment, the inflammatory environment, and downregulate inflammation.
Okay, so great. So Fred, as you mentioned earlier, the company, 301 has been evaluated in humans in an investigator-led study by Dr. Appel at Houston Methodist in 8 patients with Alzheimer's disease. Can you describe the key findings from this study?
Yeah, this was an important study because, as you mentioned, 8 patients who were mild to moderate Alzheimer's disease patients received 4 monthly cycles of the therapy, followed by 2-month post-treatment observation. And because it's very important that we always have biomarkers, where we measured Treg suppressive function, Treg numbers, as well as peripheral pro-inflammatory biomarkers, which we would expect to go down with therapy and to be consistent with what we would see clinically, measured cognitive status as well as safety. First, patients tolerated the treatment. There were no untoward effects or side effects or safety issues. What was very interesting is that on the scales that are used to measure cognition, the ADAS-Cog, CDR-SB, as well as the mini mental status exam, not only was there no progression over the course of the months of treatment, but interestingly, the mini mental status exam actually improved. So that's kind of startling, which is really important. But as I mentioned before, biomarkers are really critical in these studies. And concurrent with the improvement in the mini mental status exam, as well as the lack of progression in the other scales, we saw significant enhancement of both Treg suppressive function and Treg numbers. So that correlation with what we saw clinically was really quite important. We also saw significantly lowered plasma pro-inflammatory chemokines and cytokines in these Alzheimer patients. That also was consistent with what we would expect if we were to have an effect clinically with this therapy. We also had some imaging, and we've demonstrated on the imaging a reduction in inflammation, which again corresponds with the hypothesis. So this was well tolerated. There weren't any significant AEs. All patients completed the study, and we found this very important finding in this proof of concept study. In fact, this study was so impressive to some that the Gates Foundation decided to fund a double-blind placebo-controlled study with this therapy. And in fact, that study has just completed. And as we noted, we will report out that data in July because the last patient is completing at this point. That patient will complete sometime in March, and then we'll analyze the data and report that data. That double-blind placebo-controlled study consisted of 3 groups, 2 dose groups, one with the regimen where we saw a strong signal in the open label 8-patient study, and the other a higher dose. So one dose was 1 cycle per month, one arm was 1 cycle per month, the other arm were 2 cycles per month, and of course the third arm was placebo. And this is a 21-week treatment period study with 9-week follow-up, and the objectives are to assess safety and tolerability, biologic activity, Treg function, cytokines, chemokines, blood and CSF markers, and of course cognitive function. So this is very exciting because this is the first double-blind study using this therapy, tacking on to the proof of concept study. And we're very anxious to analyze the data, and we will report this out, as I mentioned before, this summer.
Okay, well, great. So Howard, let's go back then to the corporate strategy of using 301 as a backbone therapy and adding different elements, different agents to it. The first being 302, which is a combination product, which adds the CTLA-4 to 301, with the initial target being ALS disease. So can you further describe the mechanism of action of these 2 compounds, how they act synergistically, and why this is a good approach for ALS?
Yes. First, let me talk about the disease ALS. It's predominantly a peripheral disease, more than a central nervous system disease. And we know that whatever the underlying mechanism of the cause, whether it's dysfunctional mitochondria or the waste disposal system, inflammation is driving the disease progression. And we believe that regulatory T cells are really at the top of the pyramid that is causing that. So that by modulating Tregs, we think that we can either slow or maybe stop the disease from progressing. One of the problems in ALS is that the inflammation is so high, it's so heightened, that if you upregulate the Tregs by itself, those Tregs can become dysfunctional very quickly in that pro-inflammatory environment. So doing it just with low-dose IL-2 and propping up the Tregs, it may be okay, but it's not going to be great. And we think that, and we hypothesized, what other drugs could we combine where you can slow the inflammatory cascade and prevent those Tregs from going bad, from becoming dysfunctional. So the CTLA-4, as I mentioned before, is a way that you can have consolidative treatment therapy where you block those inflammatory mediators and those myeloid cells and those macrophages, which are key determinants of causing those Tregs to become dysfunctional. And by doing those two together, you have a synergistic effect in slowing and in propping up the Tregs and causing the functional blocks, the functional inflammatory blocks at the motor neuron to remove those functional blocks and cause hopefully the muscles to work better and the signals to fire unimpeded. So that's CTLA-4. But again, we are looking at the low-dose IL-2 in combination with many other potential drugs, and those are discussions that Arun is having, and he can further expound upon that.
Okay, so why don't we just jump to that now before we get into the data you have for 302. But Arun, what are the other possible combinations with 301 that you are exploring within your product pipeline at this time, and what are the possible indications for those different combinations?
No, thank you, Kai. At Coya, we first strongly believe that combination therapy is the future of management of neurodegenerative diseases. And I think you've heard Howard articulate this. And if you look at Coya 302 first, Kay, it is a combination of Coya 301, which is the backbone there, the low-dose IL-2 with abatacept, with the CTLA-4. You already heard from Fred that we're looking at ALS, initiating a phase 2 trial there. But just with Coya 302, we believe that there is potential in other therapeutic indications. We have publicly announced that we're going to pursue FDT, frontotemporal dementia, a disease that really has no options today and has a high unmet need. We believe the science and our dysfunctional Tregs play an essential role there, and we believe that Coya 302 has potential there. We're also going to study 302 in diseases like Parkinson's and explore and see if it would have potential there as well. So that is just on the 302 front, where 302 by itself is really a pipeline within a product. So that's a combination therapy that is a pipeline within a product. Now, if you take our low-dose IL-2 on itself, we really believe that that can serve as a backbone and be synergistic with a lot of different mechanisms to enhance efficacy in many neurodegenerative diseases. I'll just use a couple of examples here, right? If you think about the tau inhibitors or the amyloid inhibitors that are out there for Alzheimer's disease, I think the scientific rationale is there. That combination of addressing Treg dysfunction along with these mechanisms will likely be synergistic in efficacy. Not only in efficacy, but there may be potential, for example, with the amyloid inhibitors to minimize the ARIA seen as well. So we see a lot of combination potential where Coya 301, the low-dose IL-2, is the backbone upon which we build different combinations. Then there are other mechanisms like ferroptosis inhibitors that may also be amenable to combinations that are synergistic. So that's kind of why we find it very exciting, is that this platform allows us really the opportunity to pursue a lot of different indications, either in different combinations. So that's what's exciting about our portfolio. And that's where we're headed.
Well, great. So let's refocus again back on 302. Fred, 302 has also been evaluated in humans in an investigator study, again by Dr. Appel, in 4 ALS patients. So can you describe the key findings from this study?
Well, first, I'd like to step back for a moment and remind us that ALS is a very severe disease. And unfortunately, patients typically die between 2 and 5 years after diagnosis. It's a severe disease for which there are no adequate treatments. While there are approvals currently, the impact has been slight, and therefore there's a very significant unmet medical need. There's an important push by government agencies and regulatory authorities, including the FDA, to get treatments out there. And just as an example, if one looks at the PROACT ALS database, which is a very large database of patients suffering with ALS. The average rate of decline is 1 to 1.5 points per month in the scale that's used to evaluate ALS patients called the ALS-FRS. So over the span of 6 months, one would expect at least a 6-point reduction on the ALS-FRS. And that's important because 1 point, just 1 point, can be the difference between the ability to use various muscles, breathe. So 6 points is very substantial. And current therapies, while approved, don't seem to have a great impact on that 6-point reduction. So current therapies tend to also see about a 5 to 6 point reduction in a 6-month period of time. So I bring this up because there really is a significant need for treatment. And that's part of the motivator that we have to move as quickly as we are for this, with this near-term therapy that we hope to get approval in the near future. So let me talk for a moment about some of the data from the 4-patient study. These were 4 patients that included primarily sporadic type, but there was also a patient with familial or genetic type of ALS in this. These patients had baseline monthly reductions of 1 to 1.5 points per month. So they were the typical patients that one would see suffering with ALS. Patients were given this combination therapy. And the thing that I should also mention is that we have a proprietary sub-Q formulation for this combination, which is important because you want to have people to be able to self-administer or have caretakers self-administer rather than having to go into the clinic for IV treatment. So during this 6-month period where this combination treatment was given, remarkably, there was no progression or no reduction in the ALS-FRS for the first 6 months of therapy. That is remarkable. Even though it's for patients, these were well-controlled patients in terms of the evaluations, and we also, of course, measured biomarkers. And this therapy continued for a full year. And at the end of about 48 weeks, in fact, these patients had very minimal reduction. So the average across them was, excuse me, at baseline 33.5 on the ALS-FRS. After 6 months, the average was actually 33.75, essentially no change. And after 12 months, it was 32. Now, this is important because there's a relationship between the ALS-FRS and survival. So by definition, these patients will have a longer survival. At the end of the 48 weeks, the therapy was stopped, and the patients went back to their baseline progression, unfortunately. After several weeks, an extended access protocol was initiated, and the therapy was restarted. And in fact, during that time, these patients stabilized. So this is really very compelling data in these 4 patients who served as their own control. But in addition, and as I mentioned before, it's very important to measure biomarkers, chemokines and cytokines and Tregs during our studies because of our focus. And in fact, during the time of therapy, Treg suppression increased, which is what we want to suppress inflammation. Treg numbers increased, and also pro-inflammatory cytokines, chemokines decreased, and importantly, oxidative stress levels of the compounds that of what we were measuring, which included oxidative low-density lipoprotein for HNE, actually also reduced during the time of therapy. And this is very important because I'm sure you've heard of biomarkers, NFL neurofilament light, which was also measured in these patients, directionally moving as we would expect. But we also focus on these oxidative stress imbalances. Oxidative stress is essentially an imbalance between reactive oxygen species and the ability of the body to detoxify. And we found a very strong relationship between our oxidative stress measures and survival. This is very important because we believe that this correlation is even stronger than NFL. So where do we go from here? We had meetings with the FDA, pre-IND meeting, because as mentioned before, we're moving ahead with a double-blind, placebo-controlled trial with this combination therapy in ALS patients. And those meetings went extremely well. We agreed on our preclinical data, and we agreed, importantly, on a general outline of this phase 2 trial, as well as the design features that we discussed. So we have a clear path moving forward to this phase 2 study. We believe if the data from the study, which I'll describe in a moment, even comes close to what we've seen in these 4 patients, that we will be on the road towards a potential approval. And that's important to us because I described before of the significant unmet need, and we want to really move forward with providing another potential therapy for these patients. So in the first half of 2024, we are going to initiate an IND submission to be followed by a randomized double-blind placebo-controlled phase 2 trial with an open label extension. The endpoint is going to be a 6-month endpoint, the difference between the ALS FRS and placebo, and we'll follow these patients for a full year. There will be 3 groups. One group will receive the dose that the 4 patients had received. A second group will receive a higher dose, and then there will be a placebo group. The reason we're moving forward in the higher dose is that we're not just interested in reducing the progression. We are focusing on halting the progression, and we're looking to see whether that will occur with either the dose that the 4 patients were given that halted the progression in 6 months, or a higher dose, which may halt it even beyond that. This study will, again, as I mentioned before, have endpoints at 6 months and also throughout the year during the open label extension after the 6 months. And we'll be measuring cytokines, chemokines, Treg function, NFL. And importantly, our data from a registration of ALS patients that goes back decades at Houston Methodist have found some specific oxidative stress factors that correlate very closely with survival. And we're going to have separate FDA meetings to discuss validating these oxidative stress markers as a biomarker for ALS. This is very important because we will be measuring this in the double-blind study as well. And we believe we may have a significant sensitive and correlative biomarker by measuring oxidative stress markers.
So 2 follow-up questions there, Fred. First would be, in terms of the patients that you're going to enroll, I think people feel for ALS, there is a difference between fast responders and slow responders as defined by their ALS FRS scores coming into the trial. So how might you split those up? That's number one. So let me ask you that first.
So I think you were mentioning fast progressors and slow progressors.
Yes.
So
Sorry, did I say responders? Yeah.
that's fine. We're going to be focusing on the patients for which we believe will have the greatest impact of this therapy. So these are going to be patients with an ALS FRS score of at least 35 at screening. They'll have a forced vital capacity that we're comfortable with in terms of entering this study. We are going to allow patients who are the typical patients that you would see not focusing on fast progressors, because that's a patient group that's very difficult to treat, difficult to measure, and very sporadic. So we are actually going to be using guidelines to bring in patients that are the majority of patients with ALS, for which we believe will have the greatest impact.
Okay, and the second question relates to the biomarkers. It does appear that you have some, potentially very important biomarkers for oxidative stress and inflammation. Coming out of your meetings to date with the FDA, do you have a clear understanding of what you need to do to validate those, or will that take some additional communication with the agency?
Well, first, we know what we need to validate the biomarkers. We know the path that we have to take for that, but we're planning separate discussions with the FDA to validate those markers. That will serve two purposes. One, we will have a significant proprietary validated biomarker for use in ALS. That's over the longer term. But over the short term, we'll be measuring specific oxidative stress markers during our study. And of course, once you have surrogate markers, be they oxidative stress, NFL, in addition to clinical efficacy, they serve as surrogate endpoints that can allow us to progress with accelerated approvals, breakthrough designation, et cetera. So we have two tracks for the oxidative stress hormones markers. One, to correlate with the clinical efficacy we may see in our study, and two, on a separate track to validate this as a true biomarker, because I think many believe that while NFL is what we have at the current time, it's not a great biomarker with great predictive value.
Okay, great. And then just a final question for this program. What are the remaining tasks that you need to complete before you're able to file the IND?
We have a combination tox study that is starting as we speak. We don't expect any issues because we've already used this therapy separately in animal tox, and it was relatively clean without any issues. So that's something that we're starting right, as we speak, and we will have that in time for submitting the IND. So there's really very little that we need to do. We already have the protocol. We're lining up CRO, and we'll be ready to go.
Okay, great. So one of the things you mentioned earlier, Howard, was the collaboration with Dr. Reddy's. Early in 23, you had first announced that you had in-licensed their biosimilar abatacept, and then in December you announced a much more significant partnership. So, Arun, maybe you can highlight some of the key components of the agreement.
Happy to. First, I think this commercial license deal with Dr. Reddy's was a pretty transformative event for Coya. If you think about it, we executed a BD deal within the first year of being a public company. Second, I want to say that we're really thrilled to have Dr. Reddy's as our strategic partner, because we really believe in the power of 2 over 1. The two companies together bring a lot of complementary expertise and skill sets. And we're about 2 months from when we struck the deal. And I can tell you we're all cylinders firing, working extremely collaboratively. We're extremely honored to be working with Dr. Reddy's on this, advancing this program and bringing a potential product to patients fast. And that is the synergy of these kinds of partnerships. The deal itself was a $700 million plus total deal value, so a very significant deal for us. We got an upfront of $7.5 million, and all of this was in exchange for giving Dr. Reddy's the commercial rights for only one indication, ALS. Coya retains the right in all the other indications for Coya 302, and we also retain some key territories like Japan, South America, and Mexico. So we got $7.5 million in upfront, which we already have. We will, in the next few months, we should be receiving about $8.4 million once we file the IND and initiate the first dose in these patients. So events that are not too far into the future. The deal also, in addition, gives us the potential for up to $40 million in development milestones when we hit certain development milestones. In addition to that, once the product has been launched and is commercial, we're eligible for low to mid-teen royalties on all the sales in Dr. Reddy's territories, and up to $677 million in sales milestones. So this is a deal that is going to set us up really well for the future.
Great. Having done that and the other financing in Q4, Arun, so where does your balance sheet stand, coming into 24, and what's the length of the operating runway that supports?
No, absolutely. So right now we have approximately $40 million in cash. But what I also want to highlight is we are expecting to receive about $8.4 million more from Dr. Reddy's in the upcoming months. So our cash, we are right now guiding that we can get this will take us into 2026. But I again want to remind you that we have line of sight to $40 million in additional development milestones from Dr. Reddy's, assuming that we obviously meet all the milestones. So that has the potential at the right time to maybe extend our runway as well, though that is to be determined. That depends, that is contingent on us meeting certain milestones. But also what I want to say is a core part of our strategy at Coya is to continue to look for opportunities to do business development deals. Now, we're vigorous in our efforts there. As you know, BD deals, one can never guarantee what might or might not happen, but we feel pretty good based on, if you recall our previous conversation, Kay, on the potentials of the combination. I think there's a lot of opportunities with the additional indications for Coya 302, with the additional combination potential with Coya 301 for other non-dilutive business development deals. So we will always be looking for and pursuing those as well. So that is kind of where we are from a cash position and runway today.
Okay. We've had a question come in, Howard or Fred. How is your low-dose IL-2 different from the muteins that are also being developed?
Yeah, I'll take the first part, and then Fred, you can chime in. The IL-2 muteins are involved genetic modifications, amino acid changes, for instance, to make it more selective potentially for one of the receptors. And so in the case of IL-2 muteins for Tregs, they want to enhance the Tregs more selectively. Our IL-2 is a recombinant human IL-2. It's the native IL-2 that is in your body. One of the issues that we've seen with muteins, and it's actually people who have companies that have made muteins in the past, unfortunately, the efficacy has not been there in the oncology setting. And one of the problems is when you change certain parameters and you change the half-life, and you actually take away the potential of the efficacy of the protein. So the protein is meant to act very quickly. It's a short half-life. It binds to the receptor, and that's how it's meant to act. And so that's one of the limitations and issues that the muteins have. Another one is potential immunogenicity. Is there are the way that the body reacts and develops antibodies to the IL-2, the IL-2 protein, and that's another risk. And so I can tell you that the recombinant human IL-2 so far has been, well, we know it's well tolerated, and we think that this is a sort of a de-risking factor, in fact, over and above the IL-2 muteins. And I think one of the innovations that's unique about what we've done is we've taken the IL-2 recombinant and we've stabilized it in a liquid aqueous formulation at a very low dose. And that hasn't been done before. And by doing so, you're now able to, because of the short half-life, administer in the home setting. And therefore, the patients can remove it, or the caregivers can remove it out of the fridge and inject it like an insulin injection. So you overcome the issue of having patients to go into the hospital setting and intravenously administer it every single day because of the short half-life. I'll pause there. Fred, is there anything else you want to add to that?
Yeah, I would add that we've already shown that the low dose IL-2 not only enhances Treg function and number, but is tolerated without any side effects or anything of significant, any kind of clinical side effects of significance. So this is really very important. We have in our hands already a low dose IL-2 form that's safe, that does what it is intended to do. And look, this isn't rocket science. This isn't pie in the sky new mechanisms that may or may not work. This is proven technology that in our hands appears to have important efficacy signals and safety signals. And that's why we're forging ahead as we are.
Okay, great. So Arun, what are the key milestones investors should be looking for over the next 12 to 24 months?
Yeah, Kai, actually, I think what is exciting about Coya is that we have so many value-creating milestones, both short-term and mid-term. You said 12 to 24, but let me tell you the milestones that are coming up even earlier than that. In the first half of 2024, we're going to have some hopefully exciting biomarker data that we're publishing. As Fred alluded to, we'll be filing our IND to initiate this ALS phase 2 program. Two pretty critical milestones in the first half. And then early in the second half, we anticipate having the data from the Gates Foundation funded Alzheimer's study in 40 patients, study that is already fully enrolled. So that is coming up as well. So we have quite a bit of exciting milestones coming in the first half or the first part of second half of 2024 as well. And then in addition to that, we would likely file an IND for FTT in 2024 as well, and get set up for moving that into the clinic as well. And then if you look forward to 2025, we're going to have top-line results from our ALS study. That's going to be a big one. We're going to have the FTT data hopefully reading out at that time as well. So there's a lot of exciting milestones that are coming in the next 3 to 24 months for Coya. And they are in a very consistent and timely manner. We're going to see a lot of value-creating milestones. So we're very excited about what we have done as a little company here. We're very proud of our ability to execute and move things forward. So.
Okay, well, great. Well, maybe as a final question for the team, but maybe I'll start with Howard. You know, what do you think investors are underappreciating or really need to understand to fully appreciate the Coya story?
Sure. I think the first, I want to say that neuroscience now is one of the more exciting areas. It's the new oncology. There's been multiple M&A activities that have occurred recently. We think 2024 is going to be a very busy year. And I think what people should understand, and I think what you're starting to see is that combination approaches, combination therapies to target multiple pathways is the future of neurodegenerative treatments. One treatment alone is not sufficient. And because there is generally no single genetic driver of these diseases, the mechanism that we have to target the Tregs and to block and slow the other inflammatory mediators, we think is not an incremental improvement, but an exponential improvement in the way that these diseases will be handled and treated. We think that people will start to learn more about this. And in fact, Dr. Reddy's is joining our hands in getting the news out to the community and to the mainstream media. And we anticipate that that should occur very soon. And so I think as investors, I always look to, when I invest, why this company and why now? Well, we are doing some pretty remarkable, incredible things, and we've done a lot with a very little. And, I would look at a company that has past performance and success and execution. And we've done it in 2023, and I'm very bullish and confident that we're going to do the same thing, if not better, in 2024, both in the near term and the midterm and the long term. So,
yeah, thanks, Howard. Arun, anything to add?
I think Howard has said it well. What I would add is, that one thing that investors should recognize is that, one, look back at 2023, we have executed on all fronts. I think that should matter to both current and future investors. And thanks to all of our investors, because without their support, we can't be where we are. And the other point I want to again bring is that Coya 302 is a pipeline within a product. And I think that the more our investors recognize it and see that our approach has been relatively de-risked, I think they'll see the potential that this company has to be successful and to deliver.
Okay, well, great. Well, we've reached the end of our time here, but I want to thank you for everyone for joining us today. We will have a replay of the call available probably later today or tomorrow. And again, just thank you everybody for joining us today, and have a good rest of your day.
Thank you.